Hello everyone. Welcome to another talk of Speak Up Kalpana. Today we have Kaarunya Dhevi with us, who is a young scientist, and she is serving as a scientific officer in food technology division at Bhabha Atomic Research Center, also known as BARC, for the past 2 years. So, just for the background here, Bhabha Atomic Research Center is one of the premier research centers in India. And currently, Kaarunya is working on solving a major problem through her research, which is food wastage, by developing cost-effective food preservation methods. So she is also pursuing PhD alongside her full-time job as a scientific officer. So that's very interesting. I mean, she's doing both things at the same time. And she is not only good as a scientist, but also a sport person. So she was a table tennis champion during her college years. So we are very excited to have you here, Kaarunya. So it's my great pleasure to welcome you. Thank you for joining us. So now the stage is yours. So Kaarunya, you can start when you're ready.
So can you see me, Shivani?
I cannot see you.
Yes, we can.
Yeah, now I can. Yes. No.
Yeah, sure. Hi everyone, and thank you, Shivani, for a wonderful introduction. So as you know by now, my name is Kaarunya, and I'm working as a scientific officer in Bhabha Atomic Research Center. So let me just pull up my presentation. I'm speaking. Yes, so in BARC or Bhabha Atomic Research Center, my lab is a part of the food technology division, where we basically work on food irradiation. My role in this is as a food irradiation dosimetrist, which means I develop novel dosimeters for the purpose of food irradiation. And don't worry if you don't know what that means now, because I'll get into it in the talk. So I've included this slide just to give a brief idea about my background. So I hail from Coimbatore, which is a city in Tamil Nadu, and I did my schooling from Kendriya Vidyalaya, first standard to 12th standard. And in school, science was one of my favorite subjects. So after my 12, I moved to Mumbai to do integrated MSc in chemistry, and for 5 years. And in my final year, I gave the BRC entrance exam, and I cleared the exam as well as the interview. And I joined as a trainee officer. And after 1 year of training in 2018, I got posted as scientific officer. And for around 2.5 years, I've been working in BRC. So recently, actually, in January of this year, I enrolled for PhD in theoretical chemistry in the Homi Bhabha National Institute, which is a sister institute of BRC. So I'm working as well as doing my PhD right now, and I hope it goes well. So moving on to the technical part of the talk, I have 2 facts for you here. So the first one is that in terms of production of agricultural produce like cereals, pulses, fruits, and vegetables, India is among the top 3 countries. And since 2017, India stopped accepting food aid from other countries and has started exporting food, which means it's a net food exporter. On the other hand, when a country is ranked, when nations are ranked according to its potential to feed its population, India only ranks 94th out of 102 countries. And the hunger score of India is 27.2, which is serious hunger in terms of the global hunger index scale that was taken in 2020. So if you look at it in terms of percentage, around 14% of the Indian population is unnourished. Just 1 second. Yeah, around 14% of the Indian population is undernourished, and the absolute amount of people is 189.2 million, which is around quarter of the world's population. So these 2 facts, what they basically mean is that India is producing enough food, but it is not reaching everyone. And that brings into picture the concept of food security, which means enabling access of food to everyone. So what we need to do is, from the moment a food is produced, we need to reduce the food spoilage so that we can we can make sure that the food that is produced is not going to waste and it's reaching the people. So this is essentially called cutting down post-harvest losses, and the way we can do that is through food preservation. So, food spoilage is a natural thing, and there are basically four mechanisms through which it can happen. The first one is an enzymatic spoilage, which is common in fruits, and we all know that if we leave a banana outside, we don't have to do anything. No insects have to, contaminate it. It spoils on its own because it's an enzymatic process. Ripening is an enzymatic process. Similarly, if you take milk or bread, for example, bacteria and fungi, microbes, they spoil it. And if you look at pulses and rice, small insects, infestation, and pests, it's a common way of cereals and pulses getting spoiled. And chemical spoilage is the common mechanism of spoilage for foods that are high in fat, like oils and avocado. So there are already several solutions to preventing food spoilage, and some of them are well known. For example, you can just refrigerate your food, or you can heat it to kill the bacteria, or you can vacuum pack it so that you cut off the oxygen supply and you stop some of the chemical reactions or some of the uh, aerobic bacteria. Similarly, food irradiation is one such solution. It's basically a food preservation way. So, what exactly is food irradiation? In food irradiation, you expose your food item to ionizing radiation. So, ionizing radiation are a part of the electromagnetic spectrum, and they occupy the extreme right of the spectrum in this image. So, these are radiation which have energy greater than 10 kilo electron volt. So, the bond energy of most of the chemical bonds is of the order of kilo electron volt. It's also the energy through which an electron is bonded to a nucleus. So, if you expose a matter to radiation of energy greater than 10 kilo electron volt, it will invariably cause bond breakage and ionization. So, it will basically destroy the structure of any molecule that it comes in contact. So this is the mechanism through which food irradiation helps to prevent spoilage. Because in case of enzymatic spoilage, enzymes are basically proteins. It would destroy the protein structure. So whatever spoilage mechanism is in place here would basically stop. In case of microbes, it would destroy the genetic material or DNA of the microbes and kill it. Same is the case for the DNA of the eggs of the insects or the insects themselves. However, food irradiation is not a very good way of of stopping chemical spoilage, because chemical molecules are usually very small, and uh, it's the interaction probability of ionizing radiation with individual chemicals is less. So moving on, uh, what are the sources of these ionizing radiation? Where do they come from? This is where radioactivity comes into picture, and ionizing radiation are mostly byproducts of radioactive decay. So these are usually produced from radioisotopes like cobalt 60 or 137 cesium, or in the case of X-rays, these are produced through machine sources by taking an electron beam and putting a high atomic number material in front of them, and X-rays are generated. So this is the way through which ionizing radiation are produced. So I want to mention a point here. So mostly when we think of radioactivity, we think of nuclear bombs or we think of nuclear energy generation. So one of the mandates of Bhabha Atomic Research Center is to find alternate use for nuclear mm, nuclear materials or radioactive materials, and this is one such use. This is a direct societal benefit of radioactivity. So yeah, here I just want to share a very quick story on what's the benefit of doing food irradiation. So since until 1996, India used to be one of the biggest exporters, not one of the biggest, it was the biggest exporters of mangoes to countries like Australia, US, and Canada. And the trade practice at that time was that the exported items would be fumigated on arrival, which basically means they would be gassed with ethylene dibromide to kill the pests which are present in the mango. So, the common pest that was known at that time was a fruit fly, which used to lay eggs within the skin of the mango, and then the larvae would hatch and the fruits would spoil. In 1996, it was recognized globally that ethylene dibromide is carcinogenic. So, there were directives to phase out, discontinue the use of ethylene dibromide. So, when that happened, India did not have an alternate processing method to keep these pests under control. And sorry, by that time, it was also recognized that there is another pest, namely stone weevil, which used to lay in eggs within the stone, within the seed of the mango. So there was literally no way, no processing method through which you could reach inside the mango and destroy the larvae without destroying the fruit. So till 2007, there was a ban on the import of mangoes from India, and it caused some serious loss in terms of GDP for our nation that it was getting from exporting things. But in 2007, India experimented on and standardized the protocol for food irradiation of mango, and US has accepted that food irradiation can take care of these two pests. So since 2007, the export of mangoes from India has continued. So this is one of the success stories of food irradiation. Okay, so in our division, the type of research problems that we take up when related to food irradiation are these. So I've been telling you that if you supply radiation to food, you can prevent food spoilage. So how do you know how much of radiation you give to the food? So this is a topic of research, and need to do a lot of experiments, and you need to arrive at the value. Now the second point is like how do you decide, like is it random? So there are basically two factors that determine what's the amount of radiation you give to a food produce. First is pretty straightforward. You need to ensure that it is doing what you want it to do. Suppose you want to kill the insects in it, you have to ensure that you give at least that much radiation so that the insects are killed. And the second factor, which determines how much dose you give, is is that the nutritional quality and the flavor, taste, smell, etc. of the food is not destroyed or changed in an unpleasant manner because of radiation. So the minimum is decided by the effect that you want to achieve, and the maximum is capped by wanting to not have any unpleasant flavor or nutritional changes in the food. So these two are these are values that are um, products of researching food irradiation. Now, there's also another area, which is the area that I work on, is to know exactly how much radiation has been given to food. So this is a field called radiation dosimetry, dose being the technical term for how much radiation you give to food. So this is the area that I work on. Now, why is it difficult? Like, why is it challenging to find out how much radiation has been given to food? Like, can you just take an instrument and then you measure it? So there are two reasons. First is that radioactive sources, since they give out gamma rays and X rays, and I've been telling you that they kill the genetic material of insects, they destroy the structure of proteins. They can do the same thing to humans. So no one should stand near to a radioactive source without proper shielding. So doing real-time experiments by standing next to a place where irradiation is happening is completely impossible. And if you think about, if you think you can just put an instrument along with the food that is getting irradiated, okay, I'll just put it along with the food, and after it comes out of the irradiator, let me check how much dose has been given. That is also not possible because the moment you use wires or anything electronic, because of the same reason, the food does irradiation does what it does to food, that is ionization. Like the signals will go haywire because electrons will be ionized randomly from wires here and there. So you cannot use electronic instruments as well. So this leaves many. I mean, it's a challenging task. So one way of addressing it is to use a chemical system, is to use a a combination of reactants where on exposure to radiation, they would undergo a chemical reaction, and then you can measure what is the amount of product that is formed, and then you can back calculate how much radiation has been given. So this is essentially what we do in chemical dosimetry. So what I'm working on right now is called a radiochromic system, which means on exposure to ionizing radiation, a type of reaction will happen and a colored molecule will be formed. So we measure what is the amount of this colored molecule, and then we back calculate to find out how much radiation has been given. So I don't want to get into the technical details, but if you are interested, you can ask me a few questions about this after my talk. So the system that I work on, it has a dye. It's called a leukodye. Leuco means colorless. So it has a leukodye, and then it has a sensitizer. It's a sort of a, you can think of it as a catalyst sort of thing. So when these two are placed inside a radiation field, a chemical reaction will happen, and it will lead to the production of a dye, like a permanent color. So then we measure this color, and this is an organic molecule essentially what I work on, and then we measure the dose. So this is just data from one of my experiments. So what type of problems I usually face when I'm doing research of this type is that if I need to develop a system that can measure dose, I need to see that okay, it's working for the dose. What I mean is working for the amount of radiation that I intend to give to the food. So if you see the plot over here, you can see that as you're increasing the dose, you're getting an increase in the response. But sometimes what happens is you get an increase in response up to a particular dose, and then the response saturates or follows a trend that you don't really expect. When you get a graph like this, that means you can only work with this dosimeter between these two dose ranges. But if you're interested to use it for a higher dose range, you have to see what you can do, what type of combinations you can play with. Essentially, you have to optimize the system for your need. So this is something that I do. And the other thing is that it's also we also have to take into account like the economical aspects or the cost aspects, because these are all projects that would ultimately reach the food industry. So people have to use it on a regular basis. So it just cannot be at a lab scale. So if I'm doing an experiment in my lab, I will just measure the color that is formed in a spectrophotometer. But you cannot afford to have spectrophotometers in every food irradiation facility or market or wherever this thing is happening. So we need to bring down the cost. So the other thing that I'm working on right now is to see if I can use image analysis, which is basically instead of using a spectrophotometer, if I can use the camera of my phone, or if I can use a computer scanner to take an image of the dosimeter, and then if I can find out how much dose has been given. This is also something that I'm working on. So yes, so this is the end of my talk, and yeah, please feel free to ask me questions. And I want to thank the organizers for asking me to do this. And this is the first time I've been asked to talk in a place other than a scientific conference. So yeah, that's it.
So thank you, Karunya. That was amazing, a great talk. So let's see if we have any questions. So now let's move to speak up sessions. So speak up, girls, ask questions.
And you'll have to stop sharing. I'm sorry.
Yeah, Karunya, I can see myself.
Yeah.
Okay, so in the meantime, let me begin. Karunya, when you were saying that you are measuring that how much food should be radiated, how much, radiation should be provided. So is it the dosimeter which measures that?
Yeah, exactly. It's the dosimeter which measures that. Yeah.
So dosimeter is an instrument here.
It's a kind of a misnomer in the sense that it's not a instrument. It's a combination of chemicals. So it's either a solution or a like a solid tablet or something like that. So That is what is a dosimeter. It's not an instrument. So you take that and then you uh, suppose you would be irradiating your food, suppose mangoes. You put them in a
Mm-hmm.
box and push them through the irradiator. You would be
Yeah.
doing the same thing, but you would be filling the box with these small dosimeters instead of mangoes, and then you would be measuring what's the dose that they get by
Okay.
measuring the chemical change that's happening.
Okay. Okay. So we have a question here. So
Yeah,
is there no danger of food irradiation or radiation exposure?
yeah. So I'll just open my presentation just briefly because I want to show you something.
Arun, if you could also switch on your video, it would be great.
Sorry.
On your video,
Can,
that would be great.
Yeah,
your
I'll turn on my. Yeah. So if you remember, there was one slide where I was, yeah, this slide where I was saying these are the sources of radiation that are used.
I can't see your slide.
Oh, just one second. How about now?
Uh-huh. No.
Okay, fine. It's fine. I'll just uh,
But
just share.
can you turn on your video?
Yeah. Uh, can you see me?
Not now. Yeah, yes.
Yes.
Yeah. So I was mentioning in one slide that we use radioactive isotopes like cobalt and cesium and X-rays. So
Yeah.
we know that like the discovery of radioactivity itself was through other radioisotopes like radium, polonium, but we do not use those to irradiate food for the precise reason that the question that was asked, because not all sources are safe to use with food because these can induce radioactivity. So
Mm-hmm.
restriction is there's very strict restriction on what radioisotope you can use to irradiate food because you do not want to induce radioactivity. So we are using cobalt and cesium because the energy of these gamma radiation is of the range that it can never induce radioactivity. It can never cause a excitation of a nucleus. So this is the reason we use resource.
Okay, so you can measure that. Uh-huh.
Yeah.
Okay, and I'm also curious to know that. Okay, so you are working on measuring the radiation. Okay, right. So
Yeah.
right now, what food we are getting, is it radiated? Is it exposed to radiation?
So you would the it's a tricky answer because so when India exports food, it irradiates some of these food that is exported to other countries because uh, irradiated food is well accepted in certain countries like US, Canada, and all. I think
Yes.
you must be getting irradiated food in your market.
Okay.
In
Okay.
India, there's still some stigma associated with it, so uh, not everyone is convinced that it is safe. But a few food products like pet foods, for example, that we get in the
Mm-hmm.
market, these are irradiated, and also the the sachet of Maggi that we use, the spice mix,
Yes.
so
Yes.
that is also irradiated.
Okay. So spices, spice, the Maggie spice is
The Maggi
irradiated.
spice mix that you get,
Ah.
right? Yeah. So
Okay.
that is
Interesting.
also, yeah.
Yeah. Okay. So we have one more question that how much is the food wastage now? I mean, percentage of produce that goes waste.
So roughly 50% of what we produce, right? It goes to waste.
Oh, 50. Oh, so that's what when we say India is a food surplus country, but there is no enough food to provide to everyone. So that means
That
it
means
is not preserved properly.
wasted, yeah.
Ah, okay. So because of food, it is not preserved properly, so it gets wasted.
Yeah, because the food is produced at a place and then food is consumed at another place. These are separated both spatially and through time. So you have to ensure that when you bring a food from your farm to our homes, the all the supply chains on the way they have to be proper. They have to either have cold storage or some way of preserving food, and that's lacking in India because it's also an infrastructure issue. So
Okay. Okay. And you are working on that problem, that
yes,
how can we preserve food. That's
yes,
interesting. Yeah,
Yeah,
that's really interesting.
and
Okay,
that's
so
what my,
any quick, sorry.
yeah, I was saying that's what my division. That's the whole mandate of my division. And like you guys should, if you're interested, right? There's a site for BARC where we display the technologies that we, you know, develop, and it's free for public to purchase, under patents and also. So it's an
Okay.
interesting thing. You can look at it if you're interested. Yeah.
Okay. Yeah, sure, sure. Oh, I can see one hand raised. So, Gunja, you can unmute yourself and ask question.
Hello. Hello.
Yeah.
Yes. I want to ask like if we are exposed to some radiation, then there is
Yeah.
some changes or mutation in our body, DNA
Yes. Yes.
damage something kind.
Yes.
But
Yes.
the same thing when we apply on a food, of course there will be
Yeah.
some changes. How
Yes.
it
Yes.
affects our body, then how you manage that?
So interesting fact, when you cook food, right? When you're making dishes in your pan or heating, you're making dosa or something, that also causes mutation in the food. Like basically, the food that we eat, it consists of mutated, because food also has cells. Cells have DNA and everything. So the food that you eat, right, that also has mutated DNA in the sense that it's not proper DNA structure, and that doesn't do anything. So uh, it doesn't matter if you eat a DNA. It doesn't matter because it's going to your stomach. You're not, injecting it into your body or anything. So you're right that if you irradiate food, the cells inside the cells, the genetic material might be mutated, but it is safe for consumption.
And after radiation, do we do fortification of foods?
Uh, you can, if there is sufficient nutritional loss, but compared to some other processing methods, right, like heating or when you're preserving it in something, so it has been found that the nutritional loss that happens on irradiation of food is comparable. It is not too much or anything. So fortification can be done if one wishes to have an nutritionally enriched food, but it's not solely exclusive for processing by food radiation. It's also done through food that has been processed through pasteurization, etc.
Okay,
Does that
last
answer your
question.
question?
Yes, yes. And
Yeah.
last question, what are the disadvantages of radiation food?
Disadvantages. I would say the reason I was saying just now, that it is not yet started as a full-fledged food preservation practice, because we don't have enough number of irradiators yet, and it cannot be used for all foods. For example, if you irradiate fish and other high fat rich foods, right, it can cause a development of a off flavor, basically because the fats would get rancid. So it cannot be used for all types of food. So that is the disadvantage, I would say.
Okay, thank you, ma'am.
Yeah.
Okay, thanks for clarifying that, Karunya. Otherwise, everyone would have started eating less Maggie. So thanks for clarifying. It is not that dangerous.
It's
Okay.
not really that all.
Yeah. And one more thing, that so you are a chemist, okay, but many other fields can also contribute to this particular work.
Yeah, yeah,
Right. So
yeah.
how can someone from computer science background or someone from biology or physics can contribute in this kind of work?
Yeah, so biologists, I'll start with biologists because food and chemistry more closely related to food. So I was mentioning that how like we have to decide exactly how much radiation to give, and that is done after a lot of research in food irradiation. So when we need to assess if you're irradiating food and if it has made the food safe, what would you do? You would assess to see, okay, is there still some bacteria present? Like, has it reduced to a considerable level? Then you need to do microbiological studies. So biologists do that. And if you need to estimate if the nutritional loss or if there are any nutritional changes in the food after irradiation, you might need to do chemical assays as well as do some trials, probably clinical trials. So
Hmm.
there as well biologists might be involved, and if you go on the same lines, you can kind of guess that a lot of data would be involved in all this. A lot of trials, a lot of data. So we need statisticians and they are people from computer science background, they can be helpful. And physics, in fact, most of what I do, like sometimes I also need to use a lot of physics. So I did not mention this in the talk, but we use irradiators, right? So we need to simulate an irradiator to see where and all the radiation is going, because you need to have a properly shielded irradiator, and that is done through Monte Carlo simulations,
Mm-hmm.
through radiation transport codes. So I also am learning that side by side. So that is where physics people can contribute. So
Okay, interesting. So everyone, I mean, no matter what your background is, like you can be a physicist or biologist or a computer scientist or even a mathematician to solve this problem.
yeah,
So yeah, that's nice that it is very interdisciplinary field of study.
yeah, yeah.
Yeah. And Karunya, so you finished your master's and
Yeah.
now you are a scientist. So how
Yeah.
do you feel? I mean, you were a student and then right after you graduated, you became a scientist. I mean, as a scientist, how your normal day look like? What do you do?
Yeah, so one thing that was that's very different from being a student is that when you're a scientist, you straight away get your lab. Like you, I mean, it's not like you own a lab, but you have a lot of rights over a whole laboratory. That's a very big thing, right? Like I would say that's a dream. That would be a dream of a science student. You have your own lab, and yeah, you can buy instruments, but you don't have to buy them from your money. Like you can get funding from it. So that is something that I would say is a really great thing about being a scientist. Typically, yeah. So I go to lab and then I do my experiments, and sometimes we need to uh, we get visitors. We get a lot of visitors where we routinely explain about the benefits of food irradiation. So there we interact with visitors sometimes, and we do a lot of this debunking the myths of myths about food irradiation.
Mm-hmm.
So we do some outreach type of work, and as usual, like I think you must also be having a similar thing. So if we get interns, we guide them as well. So that's how my day looks normally.
Okay. So yeah, so you are enjoying your work and so what you so you did you I mean so what inspired you to become a scientist?
Science was I mean I science was just something that it was always my favorite, right? So this I just wanted to keep following it. Anything other than that just seems unnatural to me. So I would say that is what inspired me to be a scientist. But why I joined BRC, like there is a motivational factor behind it, because when I was in college, a lot of my classes were taken by some very good scientists from BRC, and I could see that I could be someone like them by joining BRC. So that's why I joined BRC. Yeah.
Uh-huh. Nice. So okay, so we have one more question. Um, is food irradiation for mangoes as effective as fumigation? And how can you compare the present techniques of food preservation to food irradiation?
Yeah. So to answer the first question, food irradiation, I would say, is better than fumigation. Uh, First one is in terms of the efficiency of pest control. Fumigation can probably deal with some of the pests that are on the superficial, on the surface, but radiation, because it can penetrate through matter, can deal with that stone beetle I was talking about. For example, it can even kill larvae or eggs that are present within the fruit. So that is in terms of efficiency of pest control also, food irradiation is better. And the second is a undisputable fact. It doesn't have any harmful effects like fumigation. You're using something that's a chemical, it's a carcinogen. Right? No matter how much you scrub, some amount of it might be left, and you're unnecessarily exposing yourself to chemicals. So that is a second way I would say. And what was the second question? I think how does food irradiation compare to other processing methods? What,
Yes. How can you compare the present techniques of food preservation, food
yeah.
preservation to food irradiation?
So comparison depends in terms of what. So I'll just say a few points for each of them. So for one was I think Gunja, right? I think she asked the same question.
Yes.
So uh, in terms of nutritional loss, uh, it's comparable, like I said in that answer. And in terms of cost, I would say it takes costs around 1 rupee to irradiate 1 kg of food. So that is in terms of cost. So depending on what are the processing techniques, they can be higher or lower. So there is that. And in terms of yeah, I think roughly it can be a complementary technique. It can be a complementary technique that's in comparison to the other techniques. And if the other techniques don't use chemicals, it's way better. That's what I would say.
Okay, but you are working on a cost-effective method, right? So that it doesn't cost more. The
Yeah,
price is same.
yeah, but what I work on is not on food irradiation as such, but overall food irradiation. I develop dosimeters. It's a subset of it. So I'm working on developing cost-effective dosimeters so that we can measure
Okay.
exactly how much food irradiation is delivering the dose.
Okay. Okay. Yeah. So let's see if we have any question from the audience. Uh, I think we are good here. So if anyone in the audience wants to connect with Karunya and wants to speak with her, so you can stay here once we end the session and directly chat with Karunya. And so Arunya, so sorry, Karunya. So so we have no more questions, and it was interesting, very nice. Thank you so much for this
Thank you.
interesting talk. So let's give a round of applause to Karunya. Yay. Thank you, Kaarunya.
Thank you. Thank you.